Hey everyone, welcome back to another episode on Chapter 3 for genetics. So far in this chapter, what we talked about was how mitosis and meiosis works. And then we talked about sex-linked inheritance. So to finish off, what we're going to do is this is going to be the final episode for Chapter 3, where it's going to be a segue into allelic interactions. To understand how allelic interactions work, you would have to understand how what gene dosage is. So gene dosage is essentially the number of genes present in the genome So essentially the way how this works is, let's assume that every gene has a certain amount of function that it gives off. So let's say gene A gives off 100% of function, so it'll be something like this. Oh, that was a bad bar graph. So yeah, let's assume that'll be sort of gene A. And we can say that for this particular gene, you would have a dominant allele, which will be big A. So big A will give that much amount of activity. But whereas if you had something like a recessive allele, like little a will give less activity. So this is basically what it's referred to as gene dosage, is that if you were to have another set of genes, like let's say an individual has a genotype of big A, little a, but let's say we added another gene A into this genotype, which will be big A, and then little a as well. So this individual will come into an issue where now you have more activity than what you were supposed to have initially. So the reason why this is important, let's assume that gene A is involved in a very complicated pathway of interaction with so many different other genes, like R, like G. And most of these individuals, they can have a diploid amount of the same gene, so 2 copies. So these 2 copies will not change that much of the pathway. But let's say you increase the amount of each particular gene. Let's say we increase big A gene by another individual, by another big A. What's going to happen is now you're going to overexpress this pathway than what you had initially, where now you're going to have a lot more big A's, and because of that big A now, you can't really digest it all to produce the little a phenotype or something like that. So that would be like the best explanation of how gene dosage works. The reason why it's important in this chapter is because X-linked X chromosomes are essentially a part of an issue on gene dosage. So as you can remember, females have 2 X chromosomes, and males also have, male only has 1 X chromosome and a Y. So the male is essentially has the correct balance of genes for the X chromosome, whereas a female doesn't. The female doesn't have the correct balance of X chromosomes because now you have 2 different X chromosomes. So essentially what happens in these cells is one of these X's, we can color them as red, and we can color them as blue. So in your cell, what will happen is that one of these X's will randomly inactivate. So let's say the blue one got inactivated, and that blue one will condense somewhere in the nucleus, and that'll be referred to as a bar body. And essentially by turning off one of your X chromosomes in females, you're essentially minimizing the gene dosage effect of having two different X chromosomes. Because you only really need just one copy of the X chromosome. So what can happen here is some phenotypes are actually the result of this X inactivation. So these are calico cats right here. And they sort of have on their fur, they have different parts of the cat where it's just randomly a different phenotype. I don't actually know, like this will be the black part and this will be the blonde part or the yellow part. So the way how that typically works is that you would have stem cells around the entire part of the fur, and then some of these stem cells can turn into, well, they can condense one X chromosome and they can condense the other. So let's say if you're big X big A, you're going to be yellow color, and then if you're X little a, you're going to be black. So let's say you have a couple of stem cells over here on the left part of your, let's make it bigger. You have a couple of stem cells on your left part of your face, and then a couple of stem cells on your right part of your face. So yes, so let's assume that the yellow one is active on this part, and then the black one, written in white, is condensed. But then the black one is activated in this stem cell randomly, and then the yellow one is condensed. So what's going to happen now is all of your cells from this point will retain that phenotype as they divide. So what happens is here now, all of these cells that come from this ancestral cell all have the yellow phenotype because they've all inactivated the other X chromosome. And then similar thing happens over here as well, where all of these ancestral cells develop that phenotype as well. So that's essentially how calico cats get their phenotype, and etc. But yeah, that's basically how that happens. And we're gonna go more into gene dosage as we go over chapter 4, which is the most interesting part of genetics in my opinion, where you start to go over allelic interactions and different types of epistatic interactions or pleiotropic interactions that play a huge role in determining how a phenotype works. But yeah, that's it.